refrigeration and freezing, using temperature to control microbial growth. Absolutely. Refrigeration, we are getting it down to not freezing. 0 degrees Celsius is freezing, that change in the state of matter from liquid water to frozen ice water, but we're not getting it down that far in refrigeration. We're getting it down to about 4 degrees Celsius is typical for your refrigeration. And then freezing is when you get below, when we change that state of matter. When we're talking about refrigeration, what we're doing there is we're just slowing down metabolism. Do you remember our metabolic growth curve? Here, I'll go back and draw it for you. So we talked about bacteria growth over time, and we said that bacteria had an optimum growth temperature. Oh, this was, yeah, temperature's over here. And this is time, and it has an optimum growth temperature. But if we get too much, I am messing this up. Here, let me try this again. We're going to put temperature here. This is the number of microorganisms on this chart, microorganisms. All right, so I've got a temperature that's my optimum growth temperature. I'm going to get the most number of microorganisms growing at this optimum growth temperature. If I get it too high, remember it falls off. I'm not going to get a lot of growth. If I get it too low, it's also going to fall off. But we talked about this in chapter 6 with microbial growth, and we said that this side, where it gets cold, we just decrease metabolism. We slow down the rate of reactions. So metabolism is slowing down here. Heat, we're denaturing stuff. We're killing stuff. We're killing stuff when we're adding the heat. Denaturing proteins, you can't come back from that. But this side of the temperature, we're just slowing down metabolism. So that's how refrigeration works. All we're doing is we're slowing down the rate of growth. So you put that pasteurized milk in there, and microorganisms are there, and they can replicate, but they're doing it slowly. So you got 2 weeks. If it's not pasteurized, you got maybe a day before those microorganisms will grow. There were more microorganisms to start with. So metabolism is still slowed down, but because we started with more with unpasteurized milk, that's the differences there. So refrigeration is just slowing down metabolism from that. When we get to freezing, freezing actually changes the state of water. So we know microorganisms need water to grow, right? I spent that whole time on metabolism. We need water to undergo cellular respiration. If we do not have water, if all of our water is ice, no growth. So that is going to stop the growth of most pathogens. So I use freezing. Again, you can keep things in the refrigerator, leftovers, maybe a week or so. In the freezer, you got longer. You've got 3 months, 6 months, maybe a year, and you can still come back from that. It's because microorganisms' growth has stopped. Long enough time usually kills microorganisms. If it's a short time, it may inhibit the growth, but it may still resume growth once you take it out of the freezer. If you leave it frozen long enough, you can kill more things. And when my kids are little in the spring, I would take a bird's nest that we found in the wintertime and I'd stick it in the freezer. And after, a couple of months in the freezer, when it came spring, I could bring that back out and the kids could touch and pull apart the bird's nest and get hands-on with the bird's nest because I killed anything that might be growing in there. So it's a good way to control microbial growth in the environment. Just physically, no chemicals needed for that. All right, so freezing. Let me talk a little bit about some different types of freezing. When I just stick something in my freezer at home, it's going to take some time before the material gets below 0 degrees Celsius. This is considered a slow freezing method. And this works, but as it's freezing slowly and that water is turning into crystals, the ice crystals are actually very sharp and they can damage cell membranes. So those ice crystals the water slowly turning into ice right next to your cell membrane are actually poking holes in your bacteria cell membrane. That's why they're dying, because you're poking enough holes in the bacteria cell membrane. So this is a slow method from that, and it usually is good for stopping things growing. So that's good. But sometimes we use freezing and we don't want to kill microorganisms or we don't want to kill cells. We can use a quick freezing technique and stop that water from making those crystals and damaging cell membranes. They still freeze, but because they're freezing so quickly, it's a different structure, and they don't damage the cells. Still some damage, but not as much as it would. Because we know freezing my chicken breast out of the freezer after 3 months or 6 months, it's not going to taste the same as when I did it fresh. There's damage. Because of those ice crystals, you're damaging proteins, you're damaging cell membranes. Okay, so let's talk about fast freezing. So say I want to preserve cell membranes. If it's a cell line in a hospital, say you've got a cancer patient and they're being treated and it's going to affect their reproductive ability. And so they say they want to freeze some cells, freezing cells in the ovaries or fertilized membranes or freezing sperm. We want that to survive freezing. We want our in vitro fertilization, where you fertilize embryos and then you take the embryos and freeze them for future attempts at implantation. We want that to survive. When we quick freeze things, we can make these cells, they're not as damaged as much and they can survive. So we use liquid nitrogen to quick freeze. Liquid nitrogen is for that. We use them for cell lines. We use it if we're shipping bacteria for our labs across we need to order more E. coli or something. We ran out. We had a mistake. We couldn't go into our lab for 3 months and all of our E. coli died and we didn't have anything frozen. We can order it and they'll ship it to us that way. That is frozen in liquid nitrogen, so we can do that. The thing you know about is, anybody gone to that, what is it, Cold Zero? What is that ice cream place over in the Vintage? You know what I'm talking about. You'd tell me if you were talking to me about it. But they use the liquid nitrogen to fast freeze the ice cream, and they say it tastes better. It's because they're not damaging those cell membranes as much. So that's what's going on there. All right, so freezing will kill more things. Slow freezing, if you're trying to kill stuff, slow freezing is the way to go. It will not sterilize. Some things can survive, and there are some pathogens that can live in colder temperatures. Listeria, most pathogens, again, freezing and refrigeration works really well. Listeria is an exception. It will actually survive lower than 0 degrees Celsius. And that's why we couldn't have Bluebell for a whole summer, because their ice cream plants got contaminated with Listeria, and they had to do the proper disinfection protocols from that. Okay, let's go back to the PowerPoint. What's next? Let's make our way through. Okay, the next section is desiccation and lyophilization. Desiccation is drying something out. So this is food dehydrators. This is, tell me this. Which would you rather, which will you be able to keep longer? Raisins or grapes? Raisins are just grapes that have been dehydrated and all the water has been removed. We know microorganisms need water. So if we remove the water, then we can keep these things longer. So controlling microbial growth in the environment, removing the water does a very good job with that. And that's how raisins work. Be careful, molds will grow on bacteria. Maybe the molds will grow on dried fruit. Maybe bacteria won't, but you do need to be careful about molds. Okay, desiccation. After desiccation is lyophilization, and this is the, have anybody been to NASA and gotten like the ice cream, the astronaut ice cream? This is where we use freezing and removing the water. This is called lyophilization. Go ahead and say that. That's a hard one. Lyophilization. And this is an example of that. If anybody backpacks or does any of this stuff, this is some lyophilized. I wish you could feel it. It's very light. This is lyophilized green beans. They removed all the water and then they froze it, and then they packaged it in this, and backpackers can take this, it's very light, you pack it in there, and open up the package and pour your boiling water right into the package, and it reconstitutes the green beans. So my husband got really into backpacking for a while, so we have a bunch of this over there, so we're not going to starve in my household, even in quarantine. And this is called lyophilization, freeze drying. And that's a method of controlling microbial growth. And in fact, on the back, it doesn't have an expiration date. It has a manufactured date, because theoretically this can last forever. It was manufactured in 2014, and it's still good now. If I poured water in this, hot water, reconstitute it, then it would be just fine for that. So controlling microbial growth does a very good job. All right, so that's lyophilization.